Assalamu alaikum everyone. So let's continue with the second part of the lecture on DNA mutations and repair. So as you probably can hear, these are the sounds of the sirens. It's Corona times. Inshallah everyone is safe and you're staying at home. So let's go over DNA repair. Now that we talked about DNA mutations, you can refer to the same pages that I told you about in the DNA mutation lecture in the Cooper's textbook. So DNA repair basically is that if there is a mutation that the cell can detect, then the cell would activate a number of pathways and mechanisms to repair the DNA. And it's really important, of course, to repair DNA because it's the genetic material, it's the hereditary material. It determines the fate, qadar, the fate of cells, whether they die or they continue to live and survive and prosper. So, there are different mechanisms that cells use to prevent DNA from mutating. And if mutated, then there are mechanisms that cells use to repair this mutation, to reverse it, or to repair the DNA. So let's talk about the first mechanism. Well, okay, so there are different mechanisms. One is prevention of errors before they happen. Then you have direct reversal of damage. You have excision repair pathways. There are different pathways or different mechanisms. You have base excision, you have nucleotide excision repair, and you have transcription-coupled repair. Then you have another mechanism known as mismatch repair. You have translesion DNA synthesis and recombinational repair. So we'll take them one by one. So let's start with prevention of errors before they happen. So basically, before anything happens to DNA, the cell removes anything that can damage DNA. Probably the most damaging effect or factor on DNA is the presence of reactive oxygen species. So these are basically molecules that contain oxygen and they are reactive. They participate in reactions. An example is oxygen radicals. These radicals are molecules containing oxygen and they are missing an electron. So they are represented this way. So you have oxygen with a lone electron. Now, this is a very reactive molecule. It's called superoxide. And it's not stable. So what it does is that it tries to attack other molecules, trying to steal electrons from them. Okay, so and this is a naturally produced molecule. So whenever there is metabolism taking place in cells, in the mitochondria, reactive oxygen species and oxygen radicals can be generated. Oxygen radicals can also be generated as a result of radiation, external radiation. So what these these molecules would do is that they need an electron badly. So they attack other molecules, lipids, sugars, proteins, DNA, RNA, whatever that is in front of them, and they steal this electron. So they oxidize molecules and they get reduced. Now, once they reduce, once this oxygen has this electron, well, it's happy, sort of. It produces hydrogen peroxide. Hydrogen peroxide is a reactive oxygen species as well, so it is reactive. And so the first reaction is, so superoxide molecules are removed by an enzyme known as superoxide dismutase. Once you have hydrogen peroxide formed, hydrogen peroxide is also removed enzymatically via an enzyme known as catalase. So catalase catalyzes one of the fastest reactions in our body. Okay, so the thing is that once this oxygen radical steals the electron from whatever molecule, the other molecule is damaged. Now, it's not really a huge problem if the damaged molecule is a protein or a sugar, because they can be regenerated. The problem is that if the damaged molecule is a lipid in the plasma membrane, then the plasma membrane is damaged, and as a result, that causes cell death. Of course, if the damaged molecule is DNA, well, that would cause mutations. It can cause breaks in the DNA, in the phosphodiester bond. It can cause depurination, or the creation or formation of AP sites, a purinic, a pyrimidinic site, and so on. So these are removed right before anything happens to DNA. Well, what if damage takes place? How can cells handle that? Well, they can reverse the damage directly. Okay, so if a damage takes place, well, this can be reversed. Now, it's really common that our skin, when exposed to UV light, to sunlight, that this UV light would hit our DNA, causing what is known as pyrimidine dimers. Okay, now the number of pyrimidine dimers formed per second is really amazing. It's like 50 to 100 reactions taking place per second once a cell is exposed to UV light. So the thing is that you have the formation of pyrimidine dimers specifically when you have two thymine nucleotides, one subsequent to the other. So you have the formation of these links, covalent links between these two thymines. Now the thing is that the structure of DNA is distorted. That is, it becomes abnormal. It's not right. And when DNA polymerase tries to synthesize DNA, or when RNA polymerase tries to synthesize RNA in transcription, well, they cannot. They stop. They stall. They stop. Okay? And that would cause, again, cells to commit suicide, die. But luckily, well, not in humans, but actually in bacteria and other organisms, but not in mammals, not in placental organisms like humans and animals. We don't have this mechanism. And the mechanism basically is that there is an enzyme known as photolyase. So what photolyase does is that it lyses, it removes the dimers, the covalent links between the thymines, reversing the thymine dimers into normal thymines. Okay, so we have other mechanisms that we utilize, that we use, specifically the nucleotide excision repair, which I will talk about in 10 minutes. But we do have a reverse mechanism, a DNA repair that is a reverse mechanism, and it's enzymatic as well. So it can happen that you have you can have methylation of guanine. We talked about methylguanine before, and we said that it pairs with a thymine rather than a cytosine. Okay, so whenever there is methylguanine in our DNA, we have an enzyme known as methyltransferase, a O6-methylguanine methyltransferase. What it does is that it removes this methyl group, it removes it to another protein, releasing it. So now the guanine becomes normal again. So we have the reverse mechanism, but it's a different type of mutation. Okay, so let's talk about the excision repair pathways, and there are different pathways that we need to talk about. We have a general excision repair pathway, we have a specific excision pathway, we have mismatch repair, and we have a coupling, a mechanism by which we can couple DNA repair and transcription together. So the general excision repair pathway, again from the name, general. So whenever there is a lesion in DNA, whenever there is modification of the bases or modification of the DNA itself, if you have the formation of pyrimidine dimers, if you have the conjugation, the attachment of a large chemical adduct. We talked about benzoapyrene in the mutation lecture. So this can be repaired using the general excision repair, which is also known as the nucleotide excision repair, NER. So what happens here in bacteria, remember, we always like to start talking about bacterial mechanisms or mechanisms, molecular mechanisms taking place in bacteria, because we understand a lot about these mechanisms. They are simpler than the mechanisms in eukaryotic cells. So what happens here is that you have activation of a complex of proteins known as UVRA. You have another protein known as UVRB, and you have a third protein known as UVRC. Each one of them has a function. They form a complex, and they are recruited. They are attracted to the lesion site. So they bind to the DNA, and they create cuts or nicks, N-I-C-K-S, nicks. Nicks means single base pair cut in the phosphodiester bond between two nucleotides. So what happens here is that they create cuts to the left and right of the lesion, and this nucleotide that contains the pyrimidine dimer is removed by a helicase, and then the gap is filled by DNA polymerase 1. Remember in the DNA replication lecture, we talked about functions of DNA polymerase 1, and one of the functions of this DNA polymerase is that it synthesizes DNA in mechanisms of DNA repair. So the gap is filled by DNA polymerase 1, and then a DNA ligase comes in and it forms a phosphodiester bond right here between the two ends of the DNA fragments. Okay. Now the thing is that I'd like to emphasize on, or I'd like to talk about, is something that is used in living organisms a lot, and that is forming protein complexes. So what happens is that to efficiently work on whatever, instead of having the three proteins separate, independent of each other, it's very common that in cells, all proteins that work on a certain mechanism, that they combine, forming a large complex of proteins, so they work together at the same time on the same place. Okay. Now in humans, the same mechanism also exists. So we do have a nucleotide excision repair pathway. We do have a similar mechanism. And this mechanism has or was identified probably in the late 1960s in people who have a condition known as xeroderma pigmentosum. It sounds like a spell from Harry Potter's movie or something. Xeroderma pigmentosum. So anyhow, so basically derma means skin, okay? Pigmentosum, pigment. So basically people with a deficiency in DNA repair, they do have this condition. Right here, for example, you have all of these pigments in a person's skin when exposed to sunlight. This is a result of cell death because of DNA damage that cannot be repaired. So it's a similar mechanism as in bacteria. The thing is that there are a number of proteins that work together, and they are known as XP proteins, and they're given a designation of A, XPA, XPB, XPC, and so on, all the way to XPG. Each one of them has a specific function. So you have proteins that can detect the damage. You can have a protein that functions as an endonuclease, making this phosphodiester bond cut on to the left and right of the lesion, you have a protein that functions as a helicase and so on. So basically, you have a number of proteins, but there are two proteins that are important that we need to talk about. One of them is actually a transcription factor. It's a protein known as transcription factor 2H. We'll talk about this protein later on in the transcription lecture. Now, this protein functions as a helicase, amazingly, huh? So what it does is that it forms a complex with all of the other proteins, and it helps in removing the nucleotide that is cut and contains the pyrimidine dimers. Okay. Another protein is replication protein A. We talked about replication protein A before. This is the single-strand DNA binding protein. This is the protein that coats the single-stranded DNA, this one right here, protecting it from degradation because cells do not like to see single-stranded DNA. Okay, they freak out. They think it's viral or something, so they start to degrade it. So anyhow, so what the replication protein A does is that it coats this DNA, protecting it from degradation, allowing for the DNA polymerase to fill in the gap, and the DNA ligase comes in, and again it forms the phosphodiester bond. Okay, so the thing is that it has also been noticed that there is a preference for repairing actively transcribed genes. Okay, in a way it makes sense. You want genes that are active to be repaired so that cells would function normally. Okay, so what happens here is that when the RNA polymerase is transcribing DNA, when it is synthesizing RNA, and there is a pyrimidine dimer, it stops, it pauses, it stalls, and this is recognized by other proteins, and they are involved. Okay, with transcription factor 2H to make an incision, make a cut to excise the DNA fragment and to fill in the gap again, so that once the gene is repaired, the RNA polymerase can bind to the gene again, and it continues with the RNA synthesis. And this actually has been identified in individuals with a condition known as Cochrane syndrome. Now, in these individuals, they have a defect in a protein known as CSB. So it's Cochrane syndrome B protein. Okay. Now, what this protein does is that it recognizes that there is a pyrimidine dimer and the RNA polymerase is not moving anymore. So once the RNA polymerase stops, CSB recognizes that, it binds to DNA, and it recruits all of the other proteins that you don't have to remember their names, but they include XPA and the replication protein A, transcription factor 2H. Again, they make a cut to the left and right. This portion is removed, and RNA and DNA polymerase fills in the gap, and then the RNA polymerase jumps on DNA and it continues with the DNA synthesis. So people with Cochrane syndrome have a number of features, including mental retardation. Anyhow, extra information. Okay. So this is the first this is the first excision pathway. Okay. We do have specific excision pathways as well. Now what happens is that you can have, again, spontaneously, or it can happen via induced mechanisms, that the base is removed, right? Creating AP sites. AP sites are apurinic sites or pyrimidinic sites. So you have a sugar, you have a phosphate, but there is no base because the glycosidic bond is cleaved. Okay, so this is recognized by an AP endonuclease repair pathway. Now, what happens here is that so you have a base that is damaged, okay, and you have this AP endonuclease repair pathway that removes the base that is damaged or that is missing a base and it can be filled in. So here's an example of a specific pathway, excision repair pathway. It's basically mediated by enzymes known as DNA glycosylases. Okay, remember how I defined enzymes like endonuclease, and I said they degrade nucleic acids in the middle. Saccharidase, they cleave or degrade sugars. Lipase, they degrade lipids, and so on. Well, DNA glycosylases, what they do is that they work on the glycosidic bond that connects the base with the sugar. So what happens is that, here's an example, it's called uracil DNA glycosylase. Now DNA should not have uracil, right? So what happens is that if there is uracil in DNA, one way or another, it doesn't matter what has induced the integration of uracil in DNA. So something happens, there's uracil in DNA, it is recognized by this enzyme, uracil DNA glycosylase. Now, what this enzyme does is that it removes uracil. So you have an AP site right here, okay? Now, what happens is that if uracil stays, that would cause a C to T transition. So instead of having a C, it converts to U. If U is not removed, then instead of having UG, you would eventually have U A, and then A, if synthesized again, a T would be replaced right here instead of U. Okay, so what happens afterwards is that you have an AP endonuclease. What it does is that it removes the nucleotide that is damaged. So this is base excision repair. So we're not removing a fragment. You're not removing a fragment. Rather, you are removing just the base that is damaged. I know my neighbors are really noisy, by the way. Anyhow, so the base is removed, and then you have the DNA polymerase comes in. It puts the right substrate in here, deoxyribonucleotide right here, and the ligase connects the fragments or the base that is added, the nucleotide that is added to the DNA fragment. Okay, so that's DNA glycosylase. This is a specific excision pathway, repair pathway. So let's say that there's a DNA mutation and it's not repaired, and the cell notices that there is, after replication, that there is something that is not right in that DNA. So we're talking about now about repair that happens after DNA is replicated. So it can happen that you can have a mismatch. That is, let's say instead of GC, you have GT, or you have AC. Well, then you have activation of a mismatch repair system. So again, it's number of proteins that work together collectively to repair this mismatch. So let's say here that you have a mismatch that's in bacteria. Again, we always start with bacteria. So you have a mismatch right here. Now you have a group of proteins known as mute proteins. Okay, mute S, mute L, mute H, whatever. All they do is the following. They make a single cut. They make a single nick right here. Okay, this is followed by the action of an exonuclease. Remember, before we talked about an endonuclease. Right here, we talked about an endonuclease because it makes a cut in the middle of DNA, right? But here, in this case, we're talking about an exonuclease that functions right there. Why? Because this point is actually, okay, let's zoom in here. This point right here is actually the end of a DNA fragment. So to remove it, you have to have an exonuclease starting right there. Okay, so you have an exonuclease that choose بمضغ بياكل the nucleotides one after the other, moving this way all the way beyond the mismatch site. Okay, So you have an exonuclease that works afterwards, and then you have a gap that needs to be filled. Of course, you have single-strand DNA binding protein protecting the single-stranded DNA, and you have DNA polymerase 1 filling in the gap, and you have a ligase that closes that. Okay, now the thing is, let's say that there is a mismatch, okay, and the mismatch is a GT. Well, both of them, G and the T, are normal looking. So how do these proteins, the mut S, mut L, mut H, know which one of them that needs to be removed? Well, there is a trick that bacterial cells do. And the trick is that DNA is methylated. That is, you have the addition of a methyl group to DNA. And this happens after replication is completed, but not immediately. Okay, so after replication is completed, what happens is that the newly synthesized strand is not methylated. So you have a strand that is methylated, that's the original strand, the parental strand, and you have a strand that is not methylated. This is the newly synthesized strand. Well, what happens is that when the mut SLH proteins come in and they see that there is a mismatch, they know which one is the original one and which one is the newly synthesized one. So what happens is that the newly synthesized DNA strand is the one that is repaired. And this methylation takes place by an enzyme known as adenine methylase. Okay, adenine methylase. So this is a nice trick. Now the thing is, do we have such a system in our cells? And the answer is yes, but of course. So yes, we do have a system that is similar in our cells, similar as in the bacterial bacterial cells, and we have two proteins known as human H, MSH2 and MLH1. These two proteins are similar to the mut S and mut L proteins in bacteria, except that we do not have an adenine methylase in our system. So how do these proteins, MSH and MLH1, how do they know which strand to repair? Well, what happens is that this happens during replication actually, and what happens is that these proteins recognize the newly synthesized strand. So they recognize that this is an Okazaki fragment because there are nicks at both ends. Okay, so right here, for example, let's focus here. So right here, you have a nick and you have another nick right here. You have a nick in here and you have a nick there. So they know that this is an Okazaki fragment, that this is a strand that is newly synthesized, and this is the one that needs to be repaired, not the opposite one. And the other one is the parental one. Okay, now that's for the Okazaki fragments. They also recognize the end of the leading strand. Okay, so they know that this is the one that is newly synthesized. This is the one that needs to be repaired, not the parental one. So that's how they recognize which strand to repair. Okay, now, are these proteins important? Very important. The reason is that there is a condition, well, we have a type of cancer known as hereditary nonpolyposis colon cancer. So this is a hereditary cancer, meaning that it is inherited, and HNPCC constitutes about 15% of colon cancer cases. So that's a lot actually. So HNPCC is actually caused by mutations mainly in MSH, and some of them are caused as a result of mutation in the MLH protein. So what happens is that people who have mutated repair systems, they are prone, they are susceptible, they have a chance of developing cancer. Because really, what is cancer? Well, how does cancer caused? Remember this, cancer is generated as a result of accumulation of genetic mutations. Okay, so what happens is that if you have a repair system that is defective, it's not functioning anymore, it cannot repair DNA, that would result in the accumulation of mutations, and some of these mutations can cause cancer. Okay, so what is cancer? Basically, it results from uncontrolled cell proliferation, cell growth, cell division. Okay, so that's colon cancer now. Well, let's say that you have a thymine dimer, a pyrimidine dimer right here, and you have DNA polymerase working on synthesizing DNA. And it hits the pyrimidine dimer. Well, how does it handle that? Well, here we have what is known as a repair system known as translesion DNA synthesis. Translesion يعني عبر الانجري, عبر الليجن. Okay, so what happens here is that the DNA polymerase stalls, it stops. We're talking about bacteria in here, right? But the same thing also exists in our system. So what happens here is that the DNA polymerase stops, it cannot continue. Now, what happens is that the DNA polymerase is released, and you have the association of other DNA polymerases like DNA polymerase 5 or 4. We talked about these briefly. We talked about these polymerases briefly in the DNA replication lecture, and all we said about them is that they are involved in DNA repair. Well, that's what they do. So what they do is that they allow for the continuation of DNA synthesis even in the presence of the lesion. Okay, so right here, so what happens is that this comes in, it jumps on the DNA, and it allows for DNA synthesis. Okay, and then it gets released, and DNA polymerase 3 comes back on, and it continues DNA synthesis. Well, what happens here? Well, what happens is that what this DNA polymerase does is that it adds any nucleotide, okay, hoping that after DNA replication continues and it's completed, that this would be repaired. Remember that cells don't like to die, okay, الروح غالية, so they like to stay alive. So what happens here is that yes, the cell is alive, it survives, except that we do have mutations that are not repaired. So the insertion of any bases would cause low fidelity. What does fidelity mean? It means like ikhlas. There is no ikhlas anymore in DNA synthesis. Okay, so especially that these DNA polymerases, DNA polymerase 4 and DNA polymerase 5, they do not have a proofreading mechanism. And as a result, this mechanism is error prone. It is associated with a high rate of DNA mutations. Okay, so it's good. We've continued DNA synthesis. It's bad. We've created even more mutations. But you know what's good as well? Is that there is a bias towards introduction of A's rather than anything whenever there is pyrimidine dimers. Okay, so since pyrimidine dimers are mainly dimers of thymine, okay, there is a preference of adding AA opposite to the pyrimidine dimer. So, you know, in a way, it's good that there is less chance that mutation is created in here. Okay, it's also something bad as well is that it's likely that C would be deaminated as well, inducing a CT transition as well. Okay, so now Let's talk about the final DNA repair mechanism, and it's known as recombinational repair. This repair pathway is important whenever you have double strand breaks. Okay, so if DNA is damaged with double strand breaks, so now both strands are broken, the phosphodiester bonds are broken. How can we fix DNA? Well, there are two mechanisms. The first mechanism is known as non-homologous end joining. So it's like grabbing both ends of this DNA and gluing them together. Okay? So you have something like this, gluing the DNA back together right here. Now that's nice, except that again it's error prone. Just gluing or connecting the ends of the DNA would create mutations. Okay. The other mechanism is known as homologous repair. What cells do is that, remember, we are diploid, right? So we have two chromosomes, one from the father, one from the father again. Let's say that the one of the parental chromosomes is broken. So you have double strand breaks. So what happens is that cells utilize the other chromosome. They take advantage of the presence of the homologous chromosome, the other parental chromosome, to fill in the gap. So you have recombination taking place, exchange of genetic material between the two chromosomes, and what happens is that the gaps are filled in by recombination, as well as DNA polymerases filling in the gap using information taken from the other chromosome. Okay, so this also illustrates what happens in the homologous repair system. Now, this needs a protein known as RAD51. Okay, RAD because it was found to be activated when cells are irradiated, when they are exposed to radiation. So this protein was called RAD51. It gets activated when cells are exposed to radiation. Now, here's something that is interesting. About 25 years ago, there was a scientist, what's her name? Her last name is King. She was working at Utah University, and she was trying to look for genes that are associated with hereditary breast cancer. That is, breast cancer that exists in families, that is inherited. And she was able to identify a gene associated with breast cancer known as BRCA1. BRCA stands for breast cancer. Later on, another protein was also identified known as BRCA2. And it has been found that both of these genes are associated with hereditary breast as well as ovarian cancers. Now, later on, when they studied the function of these proteins, they found out that both of them are involved in DNA repair. BRCA1 activates homologous recombinational repair, okay, whenever you have double-strand DNA breaks. BRCA2 is involved in recruiting RAD51 to the site of DNA break, okay? So you have again, complex of proteins working together to fix DNA, okay? Something that is of importance is that BRCA1 is also involved in transcription and transcription-coupled DNA repair as well. So this is a really an interesting protein to study, BRCA1 as well as BRCA2. Okay. Well, so this is a wrap-up. So we talked about a number of DNA repair mechanisms. So we have these four general pathways. We have the base excision repair, which is catalyzed by DNA glycosylases. They remove abnormal bases. We have the nucleotide excision repair. Specifically, they work on pyrimidine dimers or thymine dimers, or even if you have conjugation attachment of large chemical adducts like the benzoapyrene to DNA bases. And you have these proteins that are involved, the XP proteins as well as the CSB, cocaine syndrome B protein. We talked about the mismatch repair system, the involvement of the MLH1 and the MSH2, as well as the mut-S and mut-L proteins in bacteria. They work on mismatch bases, and we talked about the post-replication repair. We talked about the two pathways, homologous recombinational repair, and we talked about the non-homologous end joining and the problems associated with the non-homologous end joining repair system. Two proteins that we talked about are the BRCA1 and BRCA2, as well as the RAD51. So anyhow, we talked about a number of pathways as well, but this is sort of like a summary of what we talked about. So that's the end of this lecture. And I'd like to end by talking about this controversial issue. That is, this was a scientist who worked a few years ago who decided to actually study genes that are important for embryonic development. So she wanted to work on fertilized eggs in the early stages. So we're talking about a few days old fertilized eggs or zygote. And what she wanted to do was to manipulate, change certain genes to understand the genes, or to know, identify, and learn of the function of genes involved in early embryonic development, which is something noble. It's good. It's good to understand this because that can help us in improving in vitro fertilization, IVF. It can help us as well repair genes that are damaged and to have healthy babies. Okay, but she was like the first one in the UK to work on such topic, and it's really controversial working with zygotes. And anyhow, she got actually funding. She got the money to do what she wanted to do. I don't know what she has generated so far, but it's quite interesting. And you know, it's nice to think about these issues. What do you think about manipulating embryonic genes to have better humans? Let's say to have healthy humans. So let's say that we want to repair a specific gene so that the baby, when born, would not have a disease. What do you think about that? Is it ethical or unethical? There's a lot of talk about this issue, and in general, scientists as well as religions, they are all against doing that. But people are working on it. So it's something that will happen in the near future. And it did happen last year in China, when a Chinese scientist actually manipulated the genes of twins. So that when they are born, they would be immune. They would not be infected by the HIV virus. Okay, and they were born, and they were healthy as far as I know so far. And when he announced that to the news, it made huge, what's the word? Heated arguments about what he did, and finally. He was imprisoned actually for a year or two, I think, because he did that without having proper approval for genetic manipulation. Anyhow, think about that. We can discuss it if we meet after the corona crisis. Allah ma'akum.